This is a special episode of the Stem Cell Podcast, ISSCR 2026, Day 2. Hello, everybody. We are Doctors Daylon James and Arun Sharma. Welcome back to the Stem Cell Podcast, where we culture knowledge in stem cell research by talking to the brightest minds in the field. We're back with another special episode covering highlights from the ISSCR 2026 annual meeting in Montreal. If you're attending the conference, be sure to stop by the Stem Cell Podcast booth in the exhibit hall to meet the team, say hello, and join in on the fun. In today's episode in our series of daily recaps, we're breaking down some of the most exciting research and standout moments from day 2 of the meeting. Whether you were tied up in another session or couldn't make it to Montreal this year, we've got you covered. We're going to kick things off in just a minute, but before we get there,
did you know that you can generate more than 40 different cell types and organoids with STEMDiff HPSC differentiation kits? Explore the possibilities for reproducible, high-quality HPSC differentiation with easy-to-use serum-free kits at stemcell.com/STEMDiff.
So I'm going to kick off the daily roundup today with the first in a series. I mean, there were a handful of, well, there were a lot of reporting on clinical trial results, but a bunch of those were really focused specifically on Parkinson's disease and dopaminergic neurons in different modalities. The first was introduced by Malin Parmar, who gave the stage up to Roger Barker, who was reporting the results of the STEMPD trial, which, not coincidentally, I'm sure they had it embargoed and aligned with the drop today in the meeting, but just published today in Nature Medicine. There were these results from a series of patients in this ongoing STEMPD trial. And as I said, it's part of a wave of the PD trials that have been ongoing and that at this meeting are being reported on. The big takeaways here were, as you would expect and hope, as it relates to or compares to fetal grafts, there were no dyskinesias reported. In 6 of the 7 patients, they demonstrated clinical stability, so no worsening in the symptoms. And those patients were actually able to reduce their dose. They had PET imaging that showed important increases in dopamine output, which is key in 2 patients. And, you know, I would say an incremental advance. There was I wouldn't say pressure, but some questions from Marius Wernig at the end about this idea of a control group. And I think that's a real point of debate that the subsequent talk took up, this idea of how do you do a sham in these patients where, one, I mean, there may be some ethical considerations. And also, these patients can kind of unmask themselves from the sham group by looking by MRI to see if there's a glial scar in the path of the injection, which was a concept introduced at the end of the next speaker's talk, Jean Loring. She highlighted this as a question for the community, if we should just have an open label for these patients, because that, you know, if they can unmask themselves, what's really the point? And her talk was really about the efforts of Aspen Bioscience, which she was part of the founding group there, an analogy of the rebirth there. Aspen trees grow from the ashes, a metaphor for this regenerative field, but specifically in Parkinson's. And in this case, the real key differentiator here in terms of the modality was the use of iPS cells, right? Which is, you know, the alternative to this allogeneic, the major alternative. And what I was thinking here, and of course, there's a lot of considerations in terms of the QC, and I think Jean was really careful to highlight those and also demonstrating some really important results for 8 patients, and they have, I think, 7 subsequent patients that have also been injected with a different modality. Also no dyskinesia, also showed some improvements. But the key, I mean, what I was thinking this whole time is that while the case may have been made in terms of the rationale for using cell therapy, there's still a long way to go in terms of the scale and the economy in order to justify the value proposition. And in the same thought, I was considering that there really is such a latency when you consider the regulatory apparatus that you need to get over in order to get these therapies into people. You know, we're talking about allo and auto, you know, iPS-based therapies here. But I feel like, I don't know when it's going to be, but fast forward X years and everything's going to be hypo. And I think that's a key going to be a key milestone because of that value proposition. I think it's going to enable a real breakthrough into, scale and also just throughput. I jumped there into another talk where I caught the tail end of this story about thymus, where Jan Predimer, who is at Kyoto University, was generating thymic epithelial cells that could actually educate and generate T cells. I only caught the end of that, but it was fascinating. And that gave way to an truly enchanting talk from Miriam Mezzioni, who's a grad student at MIT. And she was speaking my language, in terms of the vasculogenesis. She used this Flick GFP model that enabled some really great imaging. It was the central question being, are there vascular progenitors in each organ? And do the vessels come from the outside in or from the outside or inside out? Identified these Wnt2 positive, Flick1 positive, EC marker negative mesenchymal progenitors that seem to be driving this phenomenon of vasculogenesis. But the key thing for me conceptually was this idea of science being recursive. Here's this grad student at the beginning of her training asking old questions with new tools. She had this great lineage tracking, live imaging. And it just really underscored for me how science is recursive. Like when I was a kid, we had these spirographs where, you're going at all these points along around the edge of this mold. But I pictured like a spirograph moving forward. We keep kind of walking in circles on this momentum forward, increasing our collective knowledge, but asking the same questions with new tools. And then I caught a really great talk, which was building on the work from Hannah of last year that he reported that ex utero development from gastrulous stage to like fetal stage mice. This is work from Alejandro Aguilera Castrejon, who was the first author on that story, now has his own lab at HHMI Janelia Farms. And he was showing how they can really carefully balance these ex utero growth environments in terms of the gases and everything, a lot of stuff, a lot of engineering there, but they could extend that window to embryonic day 12.5. And the real revelation there was that while you would expect this metabolic switch that happens during fetal stages in response to the maternal tissues, while you would expect that to be deficient in these embryos that are growing without any maternal interface, these embryo models still showed that metabolic switch. So really, I think, underscored both how much is left to be learned, how much is unknown, and how incredible these tools are for asking these questions and getting at these unexpected results. And then I jumped out of there because I couldn't miss our guy, Robert Zweigart, who is a friend of the show. And lo and behold, as I walked into the room, Arun, I saw you sitting there right in the front. Tell me, what did you think of that?
Hey, as the IPS cardiomyocyte guy, this is, one of the highlights for me was, taking these cells and two-dimensional differentiations and scaling them up like this Weigert lab is doing really well, and making mass-producing cardiac organoids literally by the billions for downstream applications for cell therapy. And that's what they were talking about, using these IPS cardiomyocyte aggregates in non-human primate cell therapy applications, right? This is a really heavy focus on process development, which was really important to me because, I'm in the process of taking a lot of my lab's work and shifting it to three-dimensional culture. So I was furiously taking notes here. His lab has done some incredible, I would say they're the world leaders in this area of suspension culture for cardiomyocytes over the past 20 years, and optimizing the large-scale cardiac organoid differentiation in these massive bioreactors, right? Multiple liter bioreactors that literally make billions and billions of cardiomyocytes. And they were using these processes to make a cardiac patch, and the patch was able to show some improvement in cardiac function in a non-human primate model. But important to note was that, and he was very upfront about this data, which I appreciated, the improvement in some of the functionality caused by the iPS cardiomyocytes was actually very clone dependent. So some of the clones had better functionality than others, but those same clones had some instances of arrhythmias that were induced. So I was happy that he was very upfront about that topic. Shifting back a little bit, I did want to talk about some of the other talks from the concurrent that I went to in the morning, which is modeling cellular complexity for tissue therapies. It was a clinical applications track. I started off with a talk from Farhane Fatahi, a new investigator at UCSF. She was modeling the body-brain axis in diseases with human pluripotent stem cells with a focus on the enteric nervous system, perhaps the forgotten child of the nervous system, but it's really pretty critical here. I mean, there's more neurons in the gut than there are in the spinal cord. I thought that was an important note to mention. And they were working on refining the human pluripotent stem cell-derived enteric ganglioid differentiation protocol to model the human enteric nervous system. And she recapped a recent Nature paper that I think we actually covered on the show that was actually showing that PP121 can direct the differentiation towards inhibitory neurons of the gut, and some really cool models of enteric neurotoxicity in an iPS chemotherapy model as well. The next talk I was at in that same session was from Dongwoo Kim, talking about PD, Parkinson's, right? This is a phase 1/2A trial of human ESC-derived dopamine progenitors in Parkinson's, application of these midbrain dopamine progenitors. And they've recently published this work in Cell Stem Cell, so we're not going to go too deep into it. But, on the same track and in the same line of what you're talking about, some of the limitations of some of these trials, this is a single center, open label phase 1/2A trial with significant immunosuppression and a low dose / high dose approach here and compassionate use and all that, right? Primary endpoints were safety and tolerability. Secondary was the efficacy. And the good news is the adverse events were largely mild and moderate. And there was actually a pretty significant improvement in motor function over 2 years, favorable trends in non-motor symptoms and quality of life as well. And one thing to mention is he said there's actually going to be a cell stem cell perspective piece in press literally today about all these different PD trials, from the Parmar group and from this group and across the world, about where they stand currently in terms of the safety and efficacy, some of the limitations, and so on. So definitely check that out if you're able to on Cell Stem Cell. Next talk was from Katja Weihnacht from Stanford, which is talking about an organ that doesn't get that much love these days. That's the thymus. She's a clinician and is focusing really heavily on diseases of the thymus, and is really interested in generating iPS-derived thymic epithelial cells to therapeutically restore immune function in the thymus. The mysterious thymus that she called it. And she also alluded to the fact that thymic health is critically important, and it's probably understated. If you have a healthy thymus, you have lower instances of cancer, better cardiovascular health, and if you actually remove and resect the thymus, you have the opposite. You have an increase in mortality from cancer, increase in cardiovascular disease, and they've had literally 10 years of work in developing this differentiation protocol from iPSC to thymus, and the hope is applying these cells first to people with congenital athymia, but eventually anybody with thymic irregularities, which, newsflash, is going to be all of us, because as you get older, which we're all getting older, you have thymic reduction in function. So keep an eye on these ITEX, thymic epithelial cells, as they call it. They're actually funded by a CERN preclinical grant to move that forward. And indeed, Robert Swigert, we already talked about his great work, and that was really exciting. And then wrapping up that session was Jean Tang, and this was one of my favorite talks for so far over these 2 days of ISSCR, because it was just so powerful. It was so emotional. It was talking about her literally multiple decades of work on the disease of epidermolysis bullosa, or EB, coming out of Stanford, where they've developed this autologous ex vivo keratinocyte sheet that's been transfected with a retrovirus expressing the COL7A1 gene for collagen, which is the gene that's messed up in EB. And long story short, they developed this clinical product called Zevaskin, which has gone through phase 1, 2, 3 over the past 10 years. And it's really just an incredible graft that's restoring quality of life in these kids and these patients who are just so suffering so badly from these lesions, causing significant itching, severe pain, and this ZevaSkin can help restore life in these people. It's really just a beautiful story. It's an amazing graft in cell therapy. Like I said, it's progressed to phase 3. The grafts still hold after many years. It took them 20 years to get to FDA approval of ZevaSkin, and she was alluding to the fact that perhaps when she comes back in 10 years, she'll have another cell therapy then, and hopefully it's not going to take less than 10 years, not the full 20, right? So that was just an amazing story, and they're working, her team is working with other folks at Stanford to develop iPSC alternatives to the cell therapy, because again, this was using primary cells, ex vivo primary keratinocytes, but that was just I think a very powerful, emotional story that I'm glad, has had a really positive result.
Yes, inspiring, heartrending glimpse of the science, but also the human dimension. I mean, those pictures, if you have kids, or maybe even if you don't, just to see it, you can't help but imagine these kids living in constant pain. So inspiring in that the progress was made, but also eye-opening view of the challenges, where you kind of have to rewrite the whole apparatus from a regulatory standpoint in order to get these therapies across the finish line. But yeah, really inspiring, and literally had me weeping into the plenary, which was also a bit somber to start, but also I think a reminder that while a big major highlight of this conference and this year is celebrating the 20-year anniversary of induced pluripotent stem cells, it's also a memorial of sorts for John Gurdon, who passed in the past year. And the plenary 2, which was chaired by Janet Rossant and Azim Surani, who were peers and colleagues and friends of the man himself, Dr. Gurdon, Sir John Gurdon. They had a sweet tribute. Janet Rossant attributed her career to him, although she had a funny joke about how she was put off by the frogs. I knew exactly what she was talking about, although I was able to tolerate it myself. And she's just such a great storyteller, has learned from so many legends. I think it really gave great closure and demonstrated, as you look at the tree, the pedigree that owes their training and their careers in many ways, their passion, to Dr. Gurdon, really underscored his impact, even across many generations. I mean, he is my own scientific grandfather. So I really felt that and was glad to see it. And that led to a talk by Elvin Bouquet, who's from Spain, about oocytes, apropos going to that first cell, which was so instrumental in Dr. Gurdon's research. But this was really a story about oocyte dormancy, how these oocytes can just chill out for decades until they're mobilized to be fertilized and become a zygote, and all the metabolic needs that are necessary for a cell that is going to lie dormant for decades. And spoiler alert, the punchline there was pyruvate, which I thought she even said herself, provided a very simple answer. But then she did admit that belied the complexity of what she has learned about metabolism in these oocytes, but also looking at it across many species, including the frog, but also the mouse and the human. So a really holistic view about this still mysterious cell that is the origin point for so much science, but also all of us. And then, I mean, I've been waiting for this, and I loved it. This is like the AI interface. They brought in Bianca Dumitrascu, who's from Columbia University. She described this programming, I guess she'd call it. It's a biophysical AI, as she called it, called Waxmorph is the title. And it's effectively in silico morphogenesis, almost philosophical in the approach, the idea being, why do organs and cells, organ primordia, take the shape that they do? How does a cell move through space? And it acknowledged a really key point, which is that this is a multi-body problem. If you guys have read the 3-body problem, which I recommend very highly, you can see when you get into higher dimensions, not just 3, but many bodies, how the permutations and all the interactions become overwhelming. And the the real key takeaway is that the cell isn't just a component of the UMAP. She used the Waddington diagram, I think, really well to describe. And if you take the UMAP from single cell, I think it's important to consider that the cell isn't just a point on those plots, but it's a free agent with programming of its own. And fundamentally, in terms of metaphor, what I saw is like the way that AI is interfacing with our society. If you have like AlphaFold, which we all know, the protein solver, if that's like the equivalent of an AI image generator, this new technology, Waxmorph, is like the equivalent of a video generator. It's taking all of these free agents in space and, has the compute to not just calculate all the interactions, but to incorporate the temporal element to calculate how organs take shape. So that really blew me away. And I thought that I could, take a nap after that. But then it was on to Sharad Ramanathan, which Arun, maybe you can take the lead there. That was another real exercise in just embryology. Again, very appropriate to the session leading off with John Gurdon. This was a real throwback. Arun, why don't you take over?
Yeah, absolutely. It was a throwback to the days of the Spearman Goldman organizer, right? Which is, a classic experiment that all developmental biologists know about, this movement of the organizer. And the tadpoles and the frog, right, where you can get multiple heads, that sort of thing, if you move the organizer correctly. It's a great connection from that classic experiment to the current era of generating more reproducible organoids and kind of the application for that old school methodology to something that we're all doing here in terms of making reproducible organoids, right? So really beautiful, simple experiments showing the importance of these morphogen-coded beads to manipulate early development. And I'm sure you can do it better justice than I can, so feel free to come back to it. But, just from my perspective, it was just a really elegant basic science study coming from the lab of Sharad Ramanathan at Harvard. And then wrapping up that particular session, that plenary 2, was Ligia Pereira talking about the Brazilian genomes. And it's almost like a Brazilian genome and a Brazilian IPS project that they've been working on for many years, exploring the vast genetic diversity in the country of Brazil, which is, a byproduct of migration, byproduct of, honestly, the slave trade over the last, centuries, and other historical events that have happened in that country to create that unique admixture in the Brazilian population. And she gave a bit of a history lesson, which I thought was actually really important in putting the work that they're doing in context. I actually didn't realize this, but Brazil has had a long track record in human embryonic stem cell work. But as is the case for a lot of IPS-derived lines in particular, the IPS generation, the iPSCs that have been generated in Brazil have largely been reflecting European ancestry, and that's true for the United States and other places around the world as well. Like I said, Brazil is a population that has significant admixture, and so an IPS bank has to appropriately reflect that genetic diversity in the country. They were taking a concept that I think Joe Wu, my PhD advisor, has talked about quite a bit, this idea of a clinical trial in a dish, and bringing it to, the country of Brazil, using, again, that population level iPSC bank, showing the genetic diversity of the country, to do a Brazilian, quote, clinical trial in a dish, right? And in general, my big takeaway for that particular talk was that, yes, greater genetic diversity is indeed needed in the IPS models. Like I said, most lines are of European ancestry. I took a bit of a lunch break, but kept on listening to some good science. Went to a Maxwell lunch showcase sponsored by Maxwell Bio, makers of multiple great MEA system, including this new high-density multi-electrode array systems that was showcased by their CSO, Marie Aubien, who I've actually gotten to know over the years because we actually have one of these systems in our own lab. And then the couple scientists who were a part of that session, this 1-hour brief lunch showcase, were Aline Martins and Hongdai Tran, who are using this MEA system for analyzing brain organoids, basically. Aline's work was actually very closely aligned to my own work because she was highlighting some space studies, actually coming from the lab of Alison Mawatri, who we've chatted with a long time ago on the podcast. They were able to grow brain organoids and organoids longer than pretty much anybody in the space stem cell field, which is over a course of 7 months in microgravity aboard the International Space Station. And we're using this HDMEA system from Maxwell to examine this long duration organoid culture. And then the second talk in that session, like I said, was from Hongdae Tran, who developed this really amazing human striatal midbrain assembloid system for modeling Parkinson's. And again, Parkinson's, once again, is a theme here in the ISSCR 2026, whether it's the clinical trials or the basic science studies. Amazing visuals about how alpha-synuclein, this protein that's thought to be, pretty heavily associated with Parkinson's, can be propagated along dopaminergic neurons, and they develop some really great visualization tools to identify that in this assembloid system. And then, moving on to the later afternoon concurrence, the 2 p.m. concurrence, I went to tissue and organoid engineering. Things were led off by Nuli Jeon from Korea, talking about his work that actually stemmed from his days back at UC Irvine in my neck of the woods about 20 years ago, showing these high throughput miniaturized vascular organoids for drug discovery, and not just blood vessels that they can generate in these high throughput NAMs, high throughput organ chip systems, but they can also generate some lymphatics as well. So it shows the malleability of that particular system. But you got to standardize the production of these chips and make them high throughput. And fast forward 20 years later, after his time at UC Irvine, that's pretty much what they've been able to do. They now have systems that enable high throughput vascularization studies and tumor angiogenesis and so on. And their systems are also really applicable to studying spheroid vascularization and using those systems for drug discovery, and actually highlighted that an IND approval was the result of some of their work that they did that was exclusively in vitro using these NAM systems, chip plus organoid. The next talk in that session was from Verena Swatch. And this is something that's, close to the work that I'm doing and the stuff that I'm interested in, which is thinking about cardiac organoids and taking them to the next level and incorporating them into this, quote, heart-brain axis, and reconstructing a human stem cell-derived neurocardiac lineage and axis on a chip. Once again, that's a great theme in this session, this NAM system and the organ-on-chip systems. They were using this to study autonomous and autonomic regulation of the human heart. These brain organoids were integrating into the cardiac organoids, and you're able to control the cardiac organoids through the functionality of the neurites extending from the brain organoids. And they had some really great videos of the neurites physically linking to the cardiac organoids. They are physically integrated, almost like a puppeteer manipulating a puppet. Really cool visual there. And they're able to show functional validation of this neurocardiac organoid with propranolol, a beta blocker. And then the last couple talks that I was able to attend in that session before I had to step out for a minute were from Asa Sherman. And he was talking about in vivo genome-wide screens of coding and non-coding genes that define the tumorigenicity of human PSCs. And then another talk from Ian Shum, he was developing a really, I think, useful system for micropatterning of high throughput, using micropatterning, UV micropatterning, for high throughput generation of embryoid bodies. And even 20 years later, embryoid bodies are still being utilized for cardiac and other differentiation protocols because sometimes they're still the most reliable. And, they're still being used to this very day. So if you want to recap the rest of that session.
Yes, that was followed by, because we were of the same mind, you and I, Arun. I was in the same session, tissue and organoid engineering, and I came there for all of that you just mentioned, but also for Sarah Zadoka, who was from Robert Zweiger's lab, who is in Robert Zweiger's lab, and because he plugged her in about, not exaggerating, is there 7, 8 people from his group presenting at the meeting, this year. He's really got a hit squad out there. And he plugged her talk because, as you alluded to earlier, there were these clones that were less effective in the transplant. And what he mentioned at the time is that they had this ex vivo tube stretching, these cardiac tubes, where they could measure the strength of the cardiac contraction of these tubes. And the clones that didn't do well in vivo also didn't do well in these ex vivo or in vitro devices. So I thought that was and he said at the time, it's a nice way of, screening clones to see which ones are going to be effective. So that's what I arrived for. And that's what she told me. Really nice, laid out the engineering involved. But what I didn't expect and was pleasantly surprised by, but there's a lot of biology to be gleaned in this system as well as a model. And what they found, effectively, a number of things. But the biggest takeaway for me was that stretch, and this is not unlike what's been reported elsewhere, but that stretch also promoted maturation of the cells, which was, I think, not a sticking point, but is a major challenge that remains in the field that's been acknowledged by all cardiac stem cell engineers and biologists. Arun, I'm sure you'll agree that maturation is an issue, but I think, coming from many angles, I think we're getting toward a solution there. And Zygarde actually made a good point. It's really important to keep in mind what you want, that there may be some benefits to transplanting fetal cells, notwithstanding their deficit in maturation. There are some edges they have in terms of engraftment. And then the highlight of that session for me was Joe P., old friend of the show, but I hadn't seen him in a while, and he came out he's rocking the full Doc Brown look. I mean, any second I expected him to say, "Where we're going, we don't need roads." And I mean, that is indicative of not just his "I don't give an F" attitude, which I've always loved about him. I mean, he does give an F. He gives a lot of Fs about the right things. And the vision, I think, is very Doc Brown, very Einsteinian, and now the hair is a match. In this one, he went over his vascularized organoid story from way back, and then he brought in a lot of new data, and I think it addressed a central point that I talked about on the show years ago. When we would talk about these vascularized assembloids, is that you can't really just take a vascular organoid and then put a organ-specific organoid next to it. The whole thing about vasculature is that it's invested, right? It's embedded, it's integrated. And he described methods that they use to generate vascularized islets, vascularized cardioids. And that's where he went real deep with the vision, and I think showed us why we all love the vision that he sets out, I think, is so out there. But then he demonstrates proof of principle. And in this case, it was heart attack on a chip, which I thought was very ambitious. But then he showed how he could recapitulate in a pretty convincing way. Very striking example of ischemic reperfusion injury in a chip. By combining immune cells from these. Previously, he's shown that you can make these heart-forming organoids that actually have a hemogenic component. So that makes these innate immune cells, and then he combines that with his vascularized organoids in this whole loop that's perfused, and he shows not only that you get this attack of the cardiac and vascular cells in the cardioid component there when it's coupled to the innate immune circulating cells, but then he uses drugs to block it, really validating the whole concept of using these kind of chips for drug discovery and validation. I think next question, everyone was scratching their head in the audience, is like, yo, have you put those drugs into the rat model yet? We'll see. Joe P., you know him, he's coming with it. If not that, then something better. But a real capstone to day 2 for me, to go back into one of the first, I think, best episodes we ever had is when Joe P. showed us a different kind of science and unleashed a wild creativity, at least in myself. Arun, he has a very conservative but directed creativity, and it served him well. We're still waiting to see how the wild creativity works out for yours truly. That's going to be it for day 2. It brings us to the end of this daily episode, but we got 2 more here at ISSCR 2026. Don't forget to follow us on X at Stem Cell Podcast or Blue Sky at stemcellpodcast.com to find out what we're up to at the meeting, and visit us at the Stem Cell Podcast booth on the exhibitor floor where you can win some prizes. We got all these pins, the pin posse. Figure out what kind of cell you are and then join a gang. And at the end of the meeting, we're gonna wow out. You'll see. Come to the booth, give you some details. Also check back here tomorrow for another episode recapping day 3 of the meeting. Until then, thank you guys so much for listening.